Let me begin by trying to put this in context. We are talking about marine primary production in the context of a planet in crisis. We need to know what's happening to our planet. We need to know what might be happening in the future and how the marine ecosystems might be affected by it. And sometimes I find that it is helpful to look away from our primary focus and look at something else. Then we can come back and see what we learned by looking elsewhere. So let's look at our neighbor, Mars. It's a red planet, very different from ours, which we are the blue planet. And over many decades, there have been many expeditions, or I shouldn't call it expeditions, but many Mars exploratory missions over the decades. And many of them have been designed to see if there is life on Mars. And how many of you have heard of Lovelock and Gaia, James Lovelock and Gaia hypothesis? Ooh, 1, 2, 3, 4. Okay, that is a gap that we need to fill. But I don't know if I have the time to talk to you a lot about him. But he is most famous for his Gaia hypothesis, where he postulated his hypothesis that our planet is a living planet that lives in harmony with its environment, and that we are in a dynamic state of balance. Anything that we do to upset that balance could have serious implications. But when he was young, he worked for NASA. And one of the things he was asked to do was to come up with an idea for looking for life on Mars. And he suggested this. He wrote about it in a 1965 paper in Nature. And he said the best thing to do is to see if the planet is in thermodynamic equilibrium. His idea was this, that if there are living organisms in the planet, and if you're looking for life on Mars, you don't know what sort of life form might be there. But whatever that might be, they will consume resources for their survival. And they will produce waste material. And since Mars didn't have oceans, the waste material would have to be deposited on the surface, or it has to appear in the atmosphere. So you look to see if the planet is in thermodynamic equilibrium. And if not, if something has been pushing it away from the equilibrium, then it means that there is something living on that planet. So that's an interesting thought. So let's compare our planet now with Mars. So if you look at the atmospheric composition of Mars and the Earth, they're very different. Mars has 96% carbon dioxide. On our planet, in our atmosphere, carbon dioxide is a trace gas. And it is changes in this trace gas that we are now concerned about. Our atmosphere has about 21% oxygen. And then remaining is 78% is nitrogen, which is inert. Now, oxygen is also in a fine balance. When I read about oxygen, some people use the word it is flammable. Some say it is not flammable, but it can help other things burn. So 21% is enough for things to burn in our atmosphere. But and 16% is necessary. But if it went much higher than that, then we would be very vulnerable to fires. So we are in a very fine balance. But the interesting thing is this. If you look back some 4.5 billion years before now, Earth looked much more like Mars. There was a lot more carbon dioxide in the atmosphere. and the Earth's atmosphere contained practically no oxygen. Then about 3.7 to 3.2 billion years ago, things started changing. And what caused this change? Experts, and I'm not one, trace this change to the arrival or to the formation on our planet of oxygen-producing cyanobacteria. So these were the first oxygen-producing photosynthesizing organisms that appeared on Earth. And in fact, they appeared in water. And things have changed dramatically since then. The oxygen in the atmosphere continued to increase, and we have finally reached the current state of about 21%. In between, during the Cambrian explosion, this is the time when the Earth went through a massive increase in biodiversity. That time is also connected with an increase in the oxygen to well beyond this 21%. So what is so special about them? The cyanobacteria and all plants and plant-like organisms since then, they contain chlorophyll. And if you were to ask me, I would say that chlorophyll is that single magic molecule that changed life on our planet. And it is what is helping us to live on this planet. No phytoplankton, no plants, no life as we know it. So now if we modify the question that NASA asked Lovelock and say, if we want to find another planet where we can live life as we live here, then I would say, look for chlorophyll. It's the single molecule that transformed our life. So what is special about chlorophyll then? What it allows the plants to do is to act as an interface, a transducer between our energy source, which is the sun, and the marine ecosystems that are built around phytoplankton. Once that link is made and energy is absorbed, then phytoplankton and all plants are able to take up. Can I do something clever? Maybe not, I won't try. It can take up water and carbon dioxide from the medium and convert it into organic material and release oxygen. And then the rest is dissipated mostly as heat. So the chemical organic material that's formed through photosynthesis becomes a form of stored energy. And that's what the rest of the marine ecosystem relies on. And on Earth, on the terrestrial, it's the same thing. Terrestrial plants are the primary producers on land, and all other organisms depend on plants directly or indirectly. So I do want to take you to the oceans. So if we are concerned about plants and forests and phytoplankton, then we have an apparent problem. It's very easy to see the forests on land. They appear green. Even when we look at, can I go back? No, so I'm going to go the other way. Yeah, if we look at the satellite image of our planet, we see that the oceans are blue, but the land, there are certain areas that are bright green, and we know that those are our lungs, the systems that keep us in oxygen. So forests on land are obvious. If you have been to coastal areas, depending on where you are, you will see macroalgae and kelp, and you'll see coastal forests. But if you look at the oceans appear for the most time, especially in the open ocean, they appear to be blue. So this might lead you to imagine that what plants are not important in the oceans, but as we know, they are, and that the oceans, wherever you go, will contain a population of phytoplankton. They may be in higher or lower concentrations or abundances. And what may not be, again, obvious is that there are thousands of species of phytoplankton in our oceans. And collectively, they do a job of fueling photosynthesis in the open ocean. And why do we not see it? It's because they are microscopic. They're very tiny, so they appear invisible to the naked eye. If you want to have some comparison, if you look at the tallest tree on land and the smallest phytoplankton in the ocean, the size difference between them is about 100 million times. So they're not so easy to see close up. But if you pull away from our planet one more time and look at the Earth from space, as this satellite is doing, say, animation from ESA, you see that as it is looking at the ocean color, there are locations that it will come to when the collective effect of the phytoplankton becomes important, and you begin to see these blooms like this one here. But it's not only the blooms, they are there in lower concentrations, and these organisms are designed, the satellites are designed to pick up small changes in the phytoplankton concentration. So the importance of phytoplankton becomes obvious if you look at the big picture. So we continue on the theme of the big picture. They are small organisms that play a big role. For if you're looking at the carbon cycle on our planet, current estimate is that phytoplankton produce some 50 teragrams, right, of carbon per year. This is roughly comparable to the net primary production by plants on land. So it's a huge amount. So if you want to now compare it with what we are doing to the planet, it is still much higher than what we are putting into the atmosphere every year. When I was very young, 1960, we were putting out about 2 gigatons of carbon per year. Now, in 2022, it's become 10. It's a staggering thought. And so all the talk that we are talking is not yet having a huge effect on how much carbon dioxide we are throwing up into the atmosphere. And the carbon dioxide concentration in the atmosphere is increasing. Here again, the oceans have a big role to play. I think I have a slide that comes up. It'll explain it a bit more. And that is this, that about, as an order of magnitude, about a third of the primary production is exported from the surface layer to the deeper waters, and in another minute fraction of it might end up reaching the bottom of the sea. And there are different processes that help this to happen. That could be direct sinking, or it could be other problems. So this serves as what is referred to as a biological pump. It's a pump that transfers energy against the gradient from the surface to the deep ocean. And when the material, in this case, organic material, leaves the surface layer of the ocean, depending on how far it sinks into the ocean and whether it is sedimented or remineralized at some depth in the ocean, the process isolates that amount of carbon from immediate contact with the atmosphere. But eventually, through remineralization and circulation and all that, it will come back to the surface layer and come once again in contact with the atmosphere. But this time scale can be from years to centuries, whereas the part of the organic material that stays in the surface layer gets recycled very fast, and the time scales are days or less to months, certainly less than a year. So phytoplankton have a big role in controlling the planetary carbon cycle. If you think about our ecosystem as a thermodynamic system, throw back to Lovelock, then you can see it as an open and dissipative system that is always losing energy perpetually. Maintaining an ecosystem is a highly energy dependent process. So the only way you can maintain that engine going is if there is a steady supply of external energy to maintain that. And that is the sun. We rely on sunlight for life. And the coupling, as I mentioned, happens through the pigment molecules and notably chlorophyll contained in phytoplankton. So it's only this very process that allows us today to talk about biogeochemical processes in the ocean. No light, no photosynthesis, no phytoplankton, you would only have geochemistry. And you may begin to look more like Mars on this planet. In the context of this training course, we are also interested in the light that leaves the ocean, because that is our signal for ocean color. So it's a two-part system. The one that leaves gives us information about what's happening in the ocean. The part that went down sustains us. So we are talking about light that is absorbed by phytoplankton. And in fact, there is a dual role for this. Not all the light that is absorbed is used for photosynthesis. In fact, photosynthesis is a minor component of the system. Most of the energy absorbed by phytoplankton is released right into the water very quickly, and it serves to localize the heating of the water through solar energy. But that's another topic. We are going to look at the minor branch of the system, which is photosynthesis. But photosynthesis itself is, to some extent at least, tied to temperature and mixing and things like that, which are related to the heating of the layers. So the idea behind this phytoplankton's role in heating is that if you have low attenuation coefficient for sunlight, or PAR in fact, then the light would penetrate deeper into the ocean, and other things being equal, mixing would be weaker and the mixed layer would be deeper. And the opposite would be true if you had a lot of phytoplankton, high mixing, shallow mixing, and light is, heating is localized closer to the surface and therefore available for exchange with the atmosphere. And these things in turn can have an effect on primary production and phytoplankton. But I did mention that the light that leaves the ocean is available to us to do ocean color remote sensing. So you can produce maps like this. This is a CWIFS map of Northwest Atlantic. We can't deny that they are very beautiful and they are appealing in that way. I'm sure we can find other things. But don't let that beauty distract you too much, because hidden in that beauty is also a lot of information. In fact, this is telling you something very fundamental about our ecosystems. Where you see a high chlorophyll concentration, that those are areas where the coupling between the energy source and the marine ecosystem, that coupling is strong. Where you have low chlorophyll concentration, that coupling is weak. So this is as fundamental as you can get And I think it is fortunate for us that we can look at this point where light and life are interacting so that we can then look at everything else that follows. Sometimes I find that in our field, because of the huge interest in what's happening to carbon, ocean color community sometimes gets apologetic about, oh, we can only give you chlorophyll, we can't give you carbon. So don't be apologetic. Tell them that chlorophyll is the most important thing that we could look at, and remote sensing is letting you do that. So as we mentioned, we need to take it beyond chlorophyll and see how we can get at primary production. But let me say this at the outset, primary production is not a direct observable from satellites. We can get at chlorophyll, that's a standard product. We can also get other things that are relevant for primary production. But primary production itself is not a direct observable. We need to calculate it, and we need to use models for that. So for those of you who are familiar with this, please forgive me for going back to the basics, but it is good to get your basics right. So let's begin by looking at what is the chlorophyll concentration. It is a measure of phytoplankton biomass. We can use other measures of biomass, can be carbon, can be ATP. You can choose whatever suits you. But phytoplankton pigment chlorophyll is also a measure of the biomass. And the index that we are going to use a lot in this talk is chlorophyll concentration. So it has dimensions of some mass per unit volume. Throughout this lecture series, if all your lecturers have been hitting the point that you need to pay attention to units. And that's very important. And I will take it a bit further. Pay attention to the dimensions. How many of you know the difference between units and dimensions? Did I see a hand up there? Would you like to explain?
So, for example, it's mass per unit volume, which is a unit, but dimension is it's a volume, it's area into say like length, distance, time. That's dimensions.
Okay. Any other? Almost right. I think you were just okay. Okay. I don't know if you got it, but the dimensions are the most fundamental properties of anything. And usually you can reduce everything that you measure to 3 fundamental units. Then other things you can derive from it, like energy, if you like. But mostly you are dealing with mass, time, and length. If you take length 3 times, you get volume. And dimensions imply just that. You say that you're measuring some length. Then you can use whatever units, as Sarita said, to quantify that length. It can be centimeter, can be inches, can be mile, can be kilometers. So if you are writing down equations, it's always good to check and balance the dimensions on 2 sides of your equation. If they don't balance, you know immediately that you have a problem, and don't go any further till you fix it. So what is primary production? It is the rate of production of phytoplankton. So therein comes the difference. Production is a rate. And if you are talking about rate, any rate that you might think of will have time in the denominator. It is a rate of change. So that means something changed per this much time. And it is important to pay attention to the time in the denominator when you are measuring production, for example. And why is that? Because you're always asking yourself, I made a measurement. When is my measurement and over what period of time is my measurement valid and useful? The answer to that lies in the time in the denominator. If your observation lasted 1 second, strictly speaking, your observation is valid for that second. If it took you a day, then it's valid for 1 day. So there is this link between the time that you spend making an observation and the time over which your measurements are applicable. And that is linked to the duration of your experiment. So this is the photosynthetic equation. I think I don't need to spend time on that because I mentioned it earlier. So looking a little bit closer at primary production, it is the rate of fixation of carbon dioxide in the presence of sunlight. And you're converting the inorganic carbon into organic matter through photosynthesis. So since it is a rate, it has dimensions of mass of carbon per unit volume, per unit time. Or if you are integrating it over the water column, then it would become mass per square length and unit time. And chlorophyll, which is a concentration, has just dimensions of meter cube. Now we have a little bit of a problem in that we want to know how much carbon was fixed. And we are trying to do most of the time that task using chlorophyll. And that is also a mass. So typically you would need to track both these masses, mass of carbon and mass of chlorophyll. Okay. So let's see, what are the growth requirements for phytoplankton? The most essential requirements come down to water and carbon dioxide. Those are the substrates on which the phytoplankton's photosystem is going to work. But then light is essential because that gives you the energy. But in addition to water, light, and carbon dioxide, they also need other elements to do their job. And these are part of the phytoplankton's architecture. And that includes nitrogen, phosphorus, iron. And many of these elements are in short supply in the surface layers of the ocean. So they can limit your primary production. So then what can make phytoplankton primary production change? We know that it changes with region and season. And these are dependent on the availability of the essential factors. So phytoplankton biomass, if you're using chlorophyll, if you want to use carbon, then carbon. That is your basic standing stock. Then the intensity and duration of sunshine, that will determine your energy supply. And the mixing in the water is also important because if the phytoplankton are being mixed vertically in the water column, depending on where you are in the water column, your light levels also change. And as I mentioned, you need the chemicals, nutrients, and temperature is considered important because they can determine the metabolic rates of metabolism of phytoplankton. And I think there's also the understanding that different types of phytoplankton react differently to environmental conditions. And so the type of phytoplankton present or the community structure can have an effect on phytoplankton. So this would tie back to what Lachlan was telling you this morning about the phytoplankton types. So once the material has been produced, what happens? Eventually, they are lost to the system. It can think of it as your capital in the bank being used or eroded. And this, the process of removal of phytoplankton could include sinking. It may be advected by horizontal currents. They may die from disease, and they may be eaten. And that's also an important pathway because all larger organisms have to derive their nutrients from phytoplankton directly or indirectly. So in the context of the computation of primary production, it's convenient to break down these factors into first order factors and second order factors. And I submit that it's convenient to think about light and pigment biomass or as the first order factors. And you can put nutrients, temperature, community structure, growth history, or photoacclimation that we've also been hearing about, and stratification, vertical mixing, things like that as secondary factors. So when you are doing a model, you can first concentrate on getting your first order principles right, and then you deal with the second order factors. So if our goal is to estimate daily water column primary production, then you can write down your problem like this. It's very general. So you want to write production P as a function of irradiance I or E. It's a choice of notation that's always problematic. Biologists like I, and for reasons that David mentioned in his talk, optical oceanographers like to use E. Anyway, let's say I for now. Sometimes you'll see I revert to E. So it's a function of light. So if you want to then express production as a function of light, then you have to express light as a function of depth, because it will vary as a function of light available at the surface, and then as a function of the depth at which you're doing your calculation. Thank you. Yeah, I'll use this one. So you want to do water column primary production where the subscripts Z and T, capital T now, indicate that you have integrated over depth and over time. And the time, if you're doing daily production, the relevant time scale is from dawn till dusk, the day length. You integrate over the day length. And in principle, you could take your depth integration to infinity, but in practical terms, it makes sense to stop at the photic depth or thereabouts. So if you have gone through these steps to calculate production at a given depth at a given time, the last step is trivial. You just sum over all the time and all the depths, and you have daily water column production. What could be more simpler than that? In fact, it gets a little bit more difficult because there are a lot of possibilities for how you might approach your model. And this is something that Trevor made where he organized all the models according to their, shall we say, complexity. So it's a kind of treatment that you want to give your data. And the way this is organized as you go from left to right and top to bottom, things get more and more I hate to use the word complex, but it gets more intricate. So in the first instance, you may simply say, I'll use a statistical regression model and be done with it. But we are not going to talk about that type of models very much any longer. But know that you can do it. You have data, you have dependent variable and independent variable, you do a regression. Or you can treat it as deterministic. And in a deterministic model, you force it with some known forcing variable, and then you can treat production as being a linear function or a nonlinear function. Again, we are going to deal mostly with the nonlinear aspects of the model. Once you have decided you want to deal with nonlinear, you have another choice. Do you deal with, how do you deal with vertical structure in the water column? You can assume that the water column is uniform, or you may accept that it has some structure in the water and that you want to account for it. Another decision is regarding whether or not you will use a spectral model or a non-spectral model. And spectral means that you're going to account for the fact that light is reaching the sea surface in a range of wavelengths. And nominally, the wavelength range from 400 to 700 nanometer is suitable for primary production. Then, for example, if you choose a spectral model, you can say, I will go with an available light model or an absorbed light model, and so on. So let's take a little bit of a closer look at these things. So one way to approach this problem is through information on the quantitative response of phytoplankton to available light. So here you have the available light I, and you have production now normalized to its unit concentration. And in these photosynthesis irradiance experiments, what you do is you incubate phytoplankton subsamples in different light levels, and you monitor primary production in each one of those containers that are exposed to different light levels. And once you have completed your experiments and you have production in each one of those light levels, you plot that on this, say in a graph, and then you fit a straight line, you fit a curve through your observations. And this is the photosynthesis irradiance curve. And if you have such a curve and you have fitted a function to it, and again, you have some options on what equation you will use to describe this curve. We all have our preferences, but anyway, many of them will do with reasonably faithful representation of your observations. So what this curve is telling you is something very important, but also very simple. If you look at the condition when light levels are very low, approaching zero, shall we say, production increases as light increases. At this light level, production is a linear function of light. And the slope of the response is the initial slope of the photosynthesis irradiance curve, or alpha B, which is a common notation for that. Then as light levels increase, the slope becomes shallower, and eventually you will reach a point when extra light is not having any effect on photosynthesis. That is supposed to represent the condition when the entire photosystem inside the phytoplankton are fully occupied, responding to the light available. So if you give it more light, it can't do anything with it. Only thing it can do is throw it out. If you continue further and give it such high levels that it begins to react badly to the extra light, you can have a third phase here, which I've not drawn, and I'm not going to dwell on too much in this talk, but we can go to it if anyone wants. You can have a decrease in production, and that stage is called the photoinhibition stage. Now, this is one of my pet topics, and I like to emphasize that. If you do models, never mind primary production, if you do any kind of models, it's always useful to differentiate between a variable and a parameter. So if you think about it, it goes like this. Let's say we have a nonlinear model of some sort. Then you have an independent variable, and something is responding to that, and that is your dependent variable. So if you have observations, you can fit a line that goes through it. The model parameters are those values of your fitted curve that distinguishes that particular curve from an entire family of curves that have the same equation. So for example, this curve here, or this curve here, or you can think of 1,000,001 curves, they can all have the same shape. But if they don't have the same parameters that determine, for example, the initial slope here and the light saturation here, you can be defining any number of these curves. So it's important to assign the parameters correctly, as well as choose your model correctly. You can have the right model, but if you drive it with the wrong parameters, you may get the wrong answer. And so if you want to throw something out because your model is not working, it would be good to decide, is my model adequate? Or is my problem that the model is fine, but I don't know the parameters? So if you are doing something like this, a nonlinear curve, simple one like this, you would require a minimum of 2 parameters to determine, to describe your curve. The parameters also have a role of describing your observations in a parsimonious, economical way, because you may have done 10 or 30 experiments to determine your curve, but 2 parameters, your initial slope and your alpha, gives you all the information about your 30 observations. Okay, so bear in mind that in this talk, your variables are light and chlorophyll and production, and your parameters are more often than not the photosynthesis irradiance parameters. But in the early part of this lecture series, you also heard from Zhongping Li, who mentioned that he prefers absorption-based models as opposed to available light models. And there are other variations on this theme. You can think about carbon-based models, and what else is there? Anyway, there are different flavors for these models. But what I would like to emphasize is that you can choose whichever model pleases you. But regardless of your choice, you really only need to have information on 4 fundamental quantities. If you have all 4 of them, then you can run your model with anything you like. And if you pay attention to the similarities between the models, you can convert from one model to the other with ease, provided you make sure that the equivalent parameters are assigned to your model. So these 4 quantities are the initial slope alpha that I mentioned. Then there is the assimilation number, or the photosynthesis, the light saturation parameter, as it's also called, which I mentioned earlier, PMB and alpha B. Then you need the specific absorption coefficient of phytoplankton. It serves you in two ways. One is to calculate light penetration in the water if you know the biomass of chlorophyll. It also helps you to convert between absorption-based and available light models. And the fourth one is the carbon to chlorophyll ratio of phytoplankton. That comes in handy if you want to use satellite data on chlorophyll, but you want to run the model as a carbon model. It also comes in handy if you want to compare your carbon production over one time step with the satellite-based chlorophyll information. Chlorophyll and carbon are not directly comparable. So if you want to go from one to the other to keep track of the two currencies, then you need to know carbon to chlorophyll ratio. It's not always needed, but it helps to be versatile. Of all these things, the carbon to chlorophyll ratio is the least well understood quantity, and that is linked to the photoacclimation that we talked about. But even in that, we are making progress. And the progress is coming through, what shall I say, a sister model to production models. And these are the photoacclimation models. And a very commonly used model is that of Geider and colleagues, and it dates back to 1997. So some of you have already shown that you know what is photoacclimation, but it works like this. I have a square or a rectangular phytoplankton cell here. I hope that's acceptable. Anyway, light is incident on the cell. And inside the cell, you have a light harvesting apparatus. And as I've been drilling in many times, that contains chlorophyll. That is what you use to harvest the light. Then there is a biosynthetic apparatus, and the available light goes to the biosynthetic apparatus, which could be the photosystem. And somewhere the cell has to decide that the energy that it received will go to produce more chlorophyll, or it will go to produce carbon and build its reservoirs of energy. So photoacclimation says that under high light conditions, it says, okay, I have a lot of light, so I can now invest my energy supply to build up my reserves. So it evolves to a level where it has low chlorophyll and high carbohydrate or whatever organic material it produces. Under the reverse condition, low light conditions, it invests in more chlorophyll production so that it can harvest the available light more efficiently, and then it invests less in the production of organic matter. This is what Geider calls the resource allocation model. Recently, we made some progress with this, and it's not that we have changed the model in any way, but with some slight replacement of variables, we were able to express photoacclimation using the same photosynthesis irradiance parameters as drive the photosynthesis. So we are now able to couple photoacclimation and photosynthesis, and the result looks like this. This is scaled irradiance. I have to go back a bit. I didn't tell you something earlier. Okay. Remember I said that we have an alpha B, an initial slope, and a light saturation parameter, PMB. Now, if you pick up this asymptotic value of PMB and draw it back to your origin where light is 0, and you also extrapolate the slope at the origin, they will meet somewhere. And you drop a line to the irradiance axis, and you will get a quantity called IK or EK. And this is often referred to as the photo adaptation parameter. It's a derived parameter and it is not independent. But bear in mind that it's derived from these two and it has dimensions of light. So if you now take the available light I and divide by IK, you get a scaled irradiance which is dimensionless. Dimensionless numbers are always nice things to find because they are often more fundamental than things that have dimensions. So anyway, if you get this one, then if you have the scaled irradiance, then you see that the carbon to chlorophyll ratio will decrease as light increases, and photosynthesis will increase as light increases. So it is this coupled process where when one goes up, the other comes down as it should. And you can drive both with the same parameters, so you have consistency across photoacclimation and photosynthesis. Now, the advantage of this is that if you have photosynthesis parameters and you know chlorophyll, you can calculate phytoplankton carbon using the photoacclimation model and chlorophyll, and you can get chlorophyll to carbon ratio. I tell you about these things because that's what I'm familiar with, but there are other ways to get at phytoplankton carbon from space. But if you have carbon from phytoplankton carbon from space, and please remember, phytoplankton carbon is not the same as POC or particulate organic carbon, but it is a component of POC. POC can contain other organic particles, maybe living like bacteria or even virus, or it may be detrital particles. Phytoplankton carbon is the living phytoplanktonic component of POC. The point I'm driving at is this, that recent developments in this area of primary production and photoacclimation have meant that you can get at both carbon and chlorophyll from space for phytoplankton, which means that you have now even more flexibility in your choice of variables to use in your model. And it also gives you an into improving your dialogue with the modeling community, ecosystem modeling community, who use typically carbon or nitrogen as their currency. So if we are able to get carbon, then we are in a better dialogue with them, opening new doors. So if that is our basic approach to computing primary production, then the next decision that I would like to discuss is whether it is worthwhile to use a spectral model rather than a non-spectral model for computing primary production. The translation between spectral and non-spectral models is very simple. If you express biomass normalized production as some function, unknown function, choose your function of choice, which contains, I see that I missed a closing brackets, never mind. You can express it as a function of this quantity capital Pi and PMB. And let me explain what I'm referring to as this capital Pi. And that is the product of light and alpha B. Regardless of which model you use, you will always find that these two quantities appear together as a product. And that is essential to balance your dimensions, because production on the left-hand side has no light in it. It is carbon per time. And light is on your right-hand side. So you have a problem. You need to get rid of light before you can have a solution that's only carbon and time. But alpha comes to your rescue. It is carbon per light available. So the light and per light gets canceled out, so you get the right dimensions. And this is the only way you can balance your dimensions. So this is how you would write a non-spectral model. And I, in this case, has no wavelength dependence in the sense that you are using the integral of the light over the photosynthetic range. It is the power, as we have all heard a lot. And remember that photosynthesis is, why should you remember? Because I didn't tell you. In case you don't know, photosynthesis is considered a quantum process. So you need to convert your light spectrum into a spectrum of photons rather than energy before you apply your photosynthesis models. Anyway, so this is power in quanta, and this is your alpha B. It's also an average value for the whole power range. If you now want to go from a non-spectral model to a spectral model, then you just have to change this quantity. And how do you change it? You accept that both light and alpha are wavelength dependent properties. So you pair them according to the wavelengths, and then you integrate them over all the wavelengths. Then the wavelength goes out, and you are back to having the right dimensions here. And if you want to use absorbed light models, then you express alpha B as a function of quantum yield, or the maximum realized quantum yield, which is the phi m here, and light absorption. Quantum yield is a measure of how much energy that is absorbed by phytoplankton is actually stored as energy. So that's your quantum yield of photosynthesis. Sometimes you'll hear of quantum yield of fluorescence. I'm not talking about that. So you can take this phi m outside and then integrate I lambda, A lambda, where A stands for phytoplankton absorption, and you are doing the same thing. Oh, sorry, I went from I to E here. Sorry, that should have been an I in this instance. So this is what you have to do. But computationally, it does add a lot of expense. In the one that we use, we use 61 wavelengths, 5 nanometer intervals from 400 to 700 nanometers. Maybe you want to use even higher. But that means that you have multiplied your computation by a factor of 60. So you have to decide if it is worthwhile, is it essential, should we do it? And you can examine that problem. Here is one way to do it. Here we used a spectral light transmission model to calculate the spectral quality of light at the base of the photic layer, regardless of what depth the photic layer is at. So what you find is that if you have very low chlorophyll concentration, let's say 0.05, phytoplankton absorption in the photic layer has very little effect on the transmitted light. So the residual light at the base of the mixed layer is blue. Whereas if you keep increasing your if you keep increasing your chlorophyll concentration, the water becomes more and more green because of the chlorophyll and auxiliary pigments present, and the residual light becomes more green. At this point, actually, it hits the wall of the high absorption by red, and you have to increase absorption by phytoplankton a lot to push it towards orange and red wavelengths. So that's a model. And here is an example of a light transmission observation in the South Pacific of Chile that Bro Johnson gave me from one of the recent expeditions to the Chilean waters. So this shows you that the light underwater does change quite dramatically depending on how much phytoplankton there is in the water. At the same time, and we have seen it repeatedly during this course, phytoplankton absorption is also strongly wavelength dependent. And that wavelength dependence can vary depending on the type of phytoplankton present. So here on the left-hand side, you have the specific absorption by phytoplankton types, going from Prochlorococcus to diatoms, chlorophytes, primnasiophytes, and so on. And you see a lot of difference. Typically, you find that small cells like Prochlorococcus would have a very high absorption in the blue and a secondary absorption in the red, both of which would be high, compared with larger cells, say diatoms, which would have a flattened absorption spectrum with lower absorption in the green and in the red, and you find all the other ones aligned in between. Now that is if you normalize it to unit chlorophyll concentration, but you can normalize it a little bit differently, such that the area under these curves are all one. That indicates how much absorption would happen if you gave it white light, just plain white light, and then you had some unknown concentration of phytoplankton, but with unit absorption spread out across the wavelengths, you would get this. But you still see that some differences remain, and the differences are again ordered in the way that they appear here. Prochlorococcus favoring blue light, diatoms relatively favoring red light, and the others are all aligned in between. This is, for me, it's interesting because if you go to the natural marine ecosystems, you will find that in oligotrophic waters, Prochlorococcus tends to dominate, especially if you are in warm waters, and high chlorophyll eutrophic waters with green light will be dominated by diatoms. And interestingly, diatoms do better in the green light than the other ones. So if you put these two together, this is what you get for absorbed light at the base of the mixed layer. The normalization is such that in a non-spectral white light environment, this product would be 1. And what you find is that when chlorophyll concentration is low, all phytoplankton absorb more efficiently and therefore have the potential for more photosynthesis. And it can be a factor of 2 here. And at the same time, when the light goes progressively green, all phytoplankton do less well, and time is running. And then diatoms do better in green light and Prochlorococcus in blue light. So if you want to include these kinds of spectral effects, then you need to include, you have to invest in a spectral model. And now, the other aspect I said we had to pay attention to was whether or not there is a vertical structure in chlorophyll concentration. One way to do that is to use a Gaussian shape, or it can be a triangle, and you describe this structure using another set of parameters of the Gaussian curve, which could be the width of the curve, the depth of the curve, and the height of the curve. And you can use satellite data to scale the value at the surface. This is, I think, the approach that Villefranche group and our own groups have followed. Some people don't explicitly have a Gaussian or any kind of deep chlorophyll maximum. So these are your options when you want to do vertical structure or not in primary production. I realized lately that the deep chlorophyll maximum is sometimes referred to as the cryptic biology. That is because if you think of an extraterrestrial, or maybe of extraplanetary origin satellite circumnavigating our planet, that satellite will not see this deep chlorophyll. So it could be confused into thinking there is no life on this planet. So it is cryptic to an extraterrestrial observer. Anyway, that's a funny thing. But so you may choose to do chlorophyll or a single peak or a double peak. And recent developments in how we parameterize these curves have led to the idea that the population of phytoplankton that exists in the mixed layer would be distinct from the one that exists below the mixed layer, and also that in low oxygen conditions, in fact, the deep chlorophyll maximum might actually appear as two peaks. So there is no direct way to get at this structure, at least as things stand now, from satellites. So if you wish to include these factors, then the only way to do it is through supplying external information. These days, with biogeochemical Argo floats providing information on chlorophyll, the potential for improving how we represent the vertical structure is improving. Um, I have to go a bit faster, but here is the point. So these are the choices that you make when you want to calculate primary production, and you are doing it at a local scale, one location, one day, and you have all the information that you need for your computations. If you want to do it globally and routinely and annually and interannually, then you are going to find yourself in a position where you have to extrapolate sparse local information to all the pixels across the entire globe to be able to do your calculation globally. So at this point, you need not only to decide on your local algorithm, and that is where you decide if you want to do linear, nonlinear, spectral, non-spectral, and all that. But you also need to establish rules for extrapolation of the local observation to large scales. That also has been preoccupying the community for a while. And there are different ways in which this problem is approached. One that we have been following is to use the biogeochemical provinces of Alan Longquist as a basis for extrapolation. And the idea is that there are these different provinces are under somewhat common forcing from external conditions. So we might apply similar rules within a province more readily than across provinces. Then there are also efforts to express variability in the parameters as a function of environmental variables, notably temperature. Two commonly used functions for expressing temperature dependence of the assimilation number is the Epley curve. And this, it makes an assumption again on what the chlorophyll, carbon to chlorophyll ratio is. And then Berenfeld and Falkowski had proposed this seventh order polynomial. And these are plotted in Heather Bowman's figure against the backdrop of observed PMB values. And in my opinion, this problem of extrapolation is the area where we really need a lot of emphasis and future research to reduce uncertainties that arise from extrapolation. Many of you know this, but if you are, and we have touched upon it at least in the earlier classes, if you are using satellite observations, you typically would use at least two wavelengths, one blue and one green, to calculate your chlorophyll values. And the diffuse attenuation coefficient changes across these wavelengths. So the satellite might be seeing a shallower depth or a deeper depth, depending on the diffuse attenuation coefficient at those depths. But if this depth is typically called the penetration depth, if it lies above the deep chlorophyll maximum, then it doesn't matter. But it does mean that you are not seeing the deep chlorophyll maximum at all. But if the deep chlorophyll maximum appears somewhere here, there could also be a distortion in your signal that the satellite receives and a residual uncertainty arising from that. So when all is said and done, and regardless of your preference for a given model or not, generally speaking, the computation of primary production can be schematized like this, where you rely on some information from the light available outside the atmosphere, and then you need to use some kind of atmospheric light transmission model to get at the surface irradiance. And these days, there are methods, satellite-based techniques that will give you surface irradiance. NASA provides that routinely. Once you know the light at the surface, you need to couple it with a light penetration model to calculate light at depth. And then you need a photosynthesis light model to say, okay, if there is so much light, how much will the production be? So these are all the models on the right-hand side. Then on the left-hand side, if you're doing it from a satellite perspective, you will get the surface biomass from satellites. And then you need to give some external information to define the biomass profile parameters and also the photosynthetic parameters. You give that to compute irradiance at depth, and you give this information to compute primary production at depth. And then you put all these things together, and you can get global primary production for whenever and wherever you want. But we do monthly these days. So if you have a long time series of these, you can, in principle, begin to address the question I started with, which was, how are the phytoplankton doing under climate change? Are they changing? Are they decreasing? Are they increasing? And I'll give you the short answer because of the time. Anyway, the answer is that the time series, the longest time series we can get from satellites, is still not sufficiently long to give a confident answer. The trends appear to be weak, except in the high latitudes where we know the environment is changing a lot more rapidly than elsewhere. And there you have these pockets of increasing trends, possibly associated with the retreating ice. And these are the inputs, the PAR and the chlorophyll. And these are some uncertainty estimates. You are assigning parameters to these calculations. So you can ask yourself what would happen if you decreased all the parameters by 1 standard deviation, or you increased by 1 standard deviation. And the answer is that these calculations can be quite sensitive to your assignment of parameters. Hence, my point that we really need to understand how these parameters behave and how they are likely to change in our environment over time with climate change. So going from computing primary production to how it is helpful in computing primary production using remote sensing, I think you can explain the utility of this by using a trivial equation. You can express the production P as a function of a biomass normalized production and a biomass value itself. So if biomass is chlorophyll, then it varies over 4 orders of magnitude. It's a highly variable quantity. And what satellites give you with unprecedented detail is information on how this biomass is changing globally, locally, seasonally. And that's invaluable information. Everything else related to the change in production per unit chlorophyll is dependent on the environmental conditions, notably light. And light also is available from satellites, at least at the surface. Then you have temperature and nutrients and so on. Either you need to now express the temperature dependent and the nutrient dependence and so on explicitly, or you can say that they are contained in the information on PI parameters that you measure in the field. So it uses all the information from the field to complement satellite data. And the satellites give you a lot of extra information that's needed to reduce uncertainties. So it uses in situ or ship observations and satellite observations as complementary tools. And you can apply it over multiple timescales. So it emerges as a method of choice. But you always need in situ observations for testing, validation, and so on. So validation of primary production is another tricky problem. Ideally, you would have two ways of measuring or computing primary production independently. Then you can compare them and establish your model performance. And this is one such example where we had actual in situ observations, and we also had PI observations, so two sets of observations of primary production at different locations, different dates. Um, and so we could use the PI parameters and the primary production model to calculate and compare it with the in situ incubated production, and the results appeared to be satisfactory. And that is when we went on to the other step. But often it is these two independent ways of measuring primary production is not available. So that becomes difficult to validate a model at the appropriate time and space scales. And another problem might be that you use all the information available to calculate your production. So you don't have independent data for validation. So in these instances, at least you can do sensitivity analyses to establish uncertainties in your model. Often, the in situ data quality might be an issue. For example, they might not have been measured under sufficient number of depths to resolve a depth-dependent production, or it might be that the incubation was only for a couple of hours, then you need to extrapolate to the whole day and so on. Then when you are trying to compare satellite-based calculations with some matched in situ observations, you run into difficulties of mismatched scales. If you're doing in vitro bottle experiments, your spatial scale at best is 1 square meter. The satellite, on the other hand, might be looking at a pixel side of 1 or 10 kilometers. So it is a 100-kilometer box, and you're comparing one observation with a 100-kilometer model, and you're bound to have mismatches. Another problem is that there are well, I don't know if I can call it indirect. There are ways of doing primary production estimates in the field that doesn't include incubations. There are field observations. You observe certain fields over time, and then you infer production from that. But such methods usually don't give you gross or net primary production. They lean towards a new production. If you're interested, we can go into those details. Another way might be to predict the biomass at some future date using your production, and then compare with what was observed by satellites at that future date. If you want to do something like that, then you have to contend with the fact that you need to convert between carbon and chlorophyll, and you also need information on all the loss terms that occurred between the two time steps. So formal error propagation or Monte Carlo methods need to be explored more. And this is the formal error calculation method that Trevor and me and others, we published in a long time ago, almost 20 years ago. So if you want to do that kind of error propagation, you first have to have some estimate of uncertainty in each of your inputs, in each of your steps. And then you compound them as in your calculation and say, okay, what is the overall error? And in our calculations, we came up with a 50% uncertainty in the precision of primary production. 50% might appear high, but remember that phytoplankton primary production varies over at least 3 orders of magnitude. So it is still very useful. The other point to remember is that this calculation is for a single observation. Satellites give you multiple observations. And if you do have multiple observations, you can use that to reduce the uncertainty in the mean that you calculate, the standard error of the mean. So if you include that, this 50% will come down quite a bit. But the trick in this case is to know how to deal with the covariance in your observations. I'll skip that because of the time. But here is my concluding slide with the key messages. At least the way we think about it, in situ databases are an essential component of doing satellite-based primary production. You need that for your sanity check and for parameterizations and a whole lot of things. We do, for example, our calculations at the global scale at monthly and 9 kilometer. Many users now say that we should go to daily 1 kilometer. And uncertainty estimations are still lacking. Where are the gaps? I think photosynthesis irradiance parameters are a big gap. And I can't resist this. I must show this. Look at this. This is our database. There's about 10,000 photosynthesis parameters. But Southern Pacific and much of the Indian Ocean remains very poorly sampled. So if you had to pick areas where we need to fill gaps, the Indian Ocean would be a key one. Yeah, and I think we are getting to this stage where it's becoming easier to compare ecosystem models and satellite-based production models. We need closer links with physical oceanographers, for example, to define phytoplankton profiles or to define the mixing and whatnot. Even to get at the mix layer depth, which is not often a reliable product. And how many of you have heard of the Global Carbon Project? Global Carbon Project? There's 1, 2, okay, 3. So there is this very high-level global carbon budget project that every year they produce a budget for the global carbon budget. But their emphasis is on what's happening in the atmosphere. The oceans are reduced to the stage where they either take up anthropogenic carbon or they release carbon to the atmosphere. They don't, in my view, they don't give even enough emphasis on what's happening in the oceans. So we need an analogous ocean carbon budget kind of initiative. And we are dealing with a highly noisy environment. How to deal with that remains a question. So if you deal with a noisy environment and you end up averaging over big time scales and spatial scales, what are you losing? The current understanding is that the phytoplankton doesn't really have a role in the air-sea carbon flux of anthropogenic carbon. I keep thinking about that and whether that assumption is right. And I think we need to really worry about the increasing occurrences of unusual algal blooms. I think we'll hear about this tomorrow or day after. Yeah, soon in this. For example, in the Arabian Sea, we've heard that large tracts of the Arabian Sea are being taken over by Noctiluca. And that's something that we should worry about. I think that is it. And okay, here is a little something for you, a gift from Bob Bruin. And that is the link to a GitHub. If you visit that, you can get a Jupyter notebook of a simple primary production model that you can play with to learn about primary production.